Welcome to Delaware Healthcare Reality, a podcast presentation of the Medical Society of Delaware with Dr. Nicholas Biasotto. Dr. Biasotto is board certified in family medicine, and he has extensive experience and training in treating patients with addiction and substance abuse. Now, let's join Dr. Nick Biasotto for Delaware Healthcare Reality.
Hi, this is Dr. Nick Biasotto, the host of the Medical Society's podcast, Delaware Healthcare Reality. And this is our 45th episode. We've done 44 that are really wonderful, and pick one or two or three and listen to them, or listen to them all. They all have some very good information for education and making you know a little bit more about what goes on in the medical field. But before we begin anything, I want to give a shout out to our sponsors, Peak Technology, the DIN, D-H-I-N, Delaware Health Information Network, Platinum Group, SourcePass, and WSFS. And anyone who has any questions, concerns, or even some suggestions for future podcasts, send your email to our email, healthcare.reality@medsocdel.org. That's healthcare.reality@medsocdel.org. We welcome your concerns and suggestions. The opinions voiced in our podcast does not reflect the opinions of the Medical Society of Delaware. They are strictly for educational and informational use only. Today we have a very special topic which is and will be cutting edge in the future of medicine and its effects on the genetics of human diseases. But before I introduce our guest, I would like to give you a brief introduction. This will help you understand what DNA is. Before the discovery of DNA, deoxyribonucleic acid, scientists knew that traits were inherited, but had no idea how these traits were passed from parents to offspring. In the 1860s, Gregor Mendel studied pea plants and established the basic laws of inheritance. Friedrich Mescher first isolated DNA in 1869 while studying white blood cells, and he found phosphorus-rich substances in the cell nuclei of the white cells. He called it nuclein, later to be called DNA. In the 1880s, again, scientists discovered a thread-like structure inside the nucleus of cells. They called this structure chromosomes, and Walter Fleming described how chromosomes behave, highlighting the role they played in heredity. In the early 1900s, Thomas Morgan performed experiments with fruit flies and discovered that their chromosomes seemed to carry the hereditary factors, and he called them genes. In the mid-1950s, convincing evidence proved DNA carried hereditary information. Then in 1953, Watson and Crick proposed the double helix of the DNA molecule, a molecule that can store information, replicate, and pass traits to one's offspring. So what is this DNA molecule? If you look at the molecule of DNA, you would see why it's called the double helix. I liken it to the spiral staircase with rungs of the staircase composed of 4 amino acids, sometimes 5, called adenosine, thymine, cytosine, and guanine. These amino acid sequences form the biological codes of life. A gene is a specific segment of the DNA staircase that carries the code for varying traits, eye color, blood type, height tendencies, and certain diseases. Humans have about 20,000 coding genes. A chromosome is an organized structure of DNA. They package and organize the DNA inside the cell nucleus. Humans have 46 chromosomes arranged in 23 pairs, a set from your mother and a set from your father. In a simplified analogy, chromosomes are the chapter of a book, genes are the sentences in the book, the instructions, and DNA are the letters of the sentences. I think I stole that from someone, but I'm not sure where, and I cannot give you a bio on that one. But today we have guests who know all about this and who are very good at what they do and have done it for many years and are on the cutting edge of getting some things improved through genetic manipulation. We have two guests today. One is Dr. Robin Miller. Dr. Miller is a nationally recognized pediatric hematologist with over 3 decades of experience in clinical care, research, and advocacy for children with blood disorders. She currently serves as director of the Sickle Cell Disease Program at the Nemours Center for Cancer and Blood Disorders at Nemours Children's Hospital in Delaware, and is a clinical associate professor of pediatrics at Sidney Kimmel Medical College of Thomas Jefferson University. She earned her undergraduate degree in biology from Johns Hopkins University and her medical degree from Temple University School of Medicine. She pediatric residency, chief residency, and fellowship in pediatric hematology oncology at Rainbow Babies and Children's Hospital at in Cleveland, Ohio. She's also pursued advanced training in clinical pharmacology and healthcare improvement. With a career defined in compassionate care, scientific rigor, and unwavering advocacy, Dr. Miller continues to lead transformative efforts to improve outcomes and quality of life for children and families affected by sickle cell anemia. Dr. Emmy Caywood is an associate professor of clinical pediatrics at the Sidney Kimmel Medical College, Thomas Jefferson University, and serves as director of transplant and cellular therapy at Nemours Children's Hospital in Delaware. She's a board-certified pediatric hematologist and oncologist, and she has led inpatient hematology, oncology, transplant, and cellular therapy services since 2014. Dr. Caywood is a principal investigator for the NCI Community Oncology Research Program at Nemours, overseeing clinical oncology research across 5 community sites in Delaware and Florida. Dr. Caywood is widely published, a committed educator, and an active leader in the National Pediatric Hematology Oncology and Transplant Collaboratives. So, how you doing, guests?
Good.
Good.
Hello.
I'd like to direct this first question to you, Robin. Exactly what is sickle cell anemia for the non-medical people who want to know what this disease is all about? And if you can, tell us what your research is doing to alter the genetic structure of the gene that carries the traits of those suffering from sickle cell disease.
So sickle cell disease is a genetic disorder that affects the beta chain of your hemoglobin. And your hemoglobin is the part of your red blood cell that carries oxygen around the body. Mm-hmm. And sickle cell happens from just one single base pair change in the beta chain DNA. And that causes your red blood cells to go from a nice round disc shape into a sickle or a C shape. And those sickle-shaped red blood cells can get stuck in the tiny little blood vessels that go to all different parts of your body. And kind of the hallmark symptom that you think of is pain. Mm-hmm. And you can get pain at any time in any part of your body. But it also can affect pretty much every organ in your body throughout your life. So it's a chronic, lifelong disease. And it affects predominantly people of African origin. So in the United States, it's mainly African Americans, but also Hispanic people, people from certain parts of Asia, and there's about 100,000 people in the United States with sickle cell, mm-hmm. but the vast majority are from sub-Saharan Africa.
So what are you doing to cure this disease?
Yeah,
if that is
so
possible?
That's a big question.
Mm-hmm. I think it might be helpful to start with what current treatments are for sickle cell disease.
Good. Let's do that. Thank you, Emmy.
So currently we have treatments to treat the symptoms and to address complications when they happen. So we use strong pain medicine for the pain crises. Often patients end up in the hospital on high doses of opioid pain medicine like morphine. And we do have blood transfusions actually are a mainstay of treatment for sickle cell complications. And some people, maybe about 10 or 20%, have to get regular blood transfusions to keep them healthy. And there are also some disease-modifying drugs, and the one that we've had the longest and has the most data behind it is a medicine called hydroxyurea that you have to take every day to help prevent pain and some of the other complications. And it helps a lot, mm-hmm. and it can improve the length of life, but it's by no means a home run, and it doesn't cure the disease.
Okay. So, now there are approaches that we're looking for these individuals to possibly offer them a cure or a significant modification of their disease disorder. One is the bone marrow and stem cell transplantation type, and that's where I'm going to address this to you, Emmy. Tell me a little bit about that. How is it done, as simply as you can?
So bone marrow transplant was developed approximately 40 years ago, really as a method to treat disorders of the bone marrow, namely leukemias, which are a cancer of the bone marrow, and other inherited bone marrow failure disorders. Mm-hmm. It was noted in the 1980s that a child with sickle cell disease, who unfortunately also developed leukemia, was cured of both her leukemia and her sickle cell disease by undergoing a bone marrow transplant.
Very interesting, fascinating.
And since that time, bone marrow transplant has been used as a cure for patients with severe sickle cell disease. So because I'm a pediatric transplanter, I see mainly pediatric and young adult patients. Those are the patients referred to me by Robin and her team who have unusually severe sequelae or symptoms of their disease. Okay. So have suffered a stroke or have developed something called alloimmunization, which makes it very difficult to receive transfusions to ameliorate symptoms.
Okay.
So we have used transplant in those cases when we have a well-matched donor.
Gotcha.
With good success, but it's a risky procedure.
Because
of the therapies that are needed in order to accomplish a transplant.
One of those therapies is chemotherapeutic destruction of the bone marrow, right?
So traditionally we have needed to use high doses of chemotherapy to make room in the bone marrow for a donor's bone marrow cells. Mm-hmm. The other major complication of bone marrow transplant, especially for this patient population, has been to find an adequately well-matched donor. Mm-hmm. Because sickle cell disease is a genetic disease, it limits the number of potential donors for any given patient with sickle cell. There's only a 25% chance that any sibling is going to be a full match, and patients with a full match donor tend to do the best.
Great. But it's kind of rough, isn't it, when you have to destroy the bone marrow and you have to keep the person in a sterile environment prior to the transplant, correct?
It's a blunt instrument to change a single gene disorder.
Interesting.
Yeah.
That's a good way to put it. So the pluses are potential cure, right? Potential. But as you mentioned, the limitations are you need a close match, and you got to worry about what the chemotherapeutic effects on the bone marrow are going to do to the person, obviously. And, there's always the risk of a donor match, a donor and host problem with the match, correct?
Mm-hmm. Yeah. One of the major complications of a traditional bone marrow transplant is a reaction called graft versus host disease. Right. This is a disorder that can affect any organ in the body, and is basically mediated by the immune system identifying a recipient's body as the wrong place to be. And when the immune system identifies a recipient's body as the wrong place to be, it causes destruction. And the last thing we want to do is substitute one severe disorder, sickle cell disease, for another disorder, graft versus host disease.
Understandable. Well, we have another method, that gene therapy and editing. I'm going back to you, Robin. How about you discuss a little bit of that research that's going on in that regard?
But before I go there, I just wanted to add a little something
Sure,
to
go
what
right
Emmy
in.
said, that even though bone marrow transplant with the donor being a completely matched sibling has an extremely high success rate, like better than 90 or 95% in most cases for cure, because of all the things that you talked about, what the patients have to go through to get there. The chemotherapy causes a lot of side effects, including infertility. It's a really hard decision for families to make, because whereas if you're getting a bone marrow transplant for leukemia, usually your chances of dying from the leukemia in the short term without the transplant are very high.
Right.
But for sickle cell disease, it's a lifelong chronic disease with lots of complications
Lots
and
of
illnesses.
suffering, too. Mm-hmm.
Yeah, suffering. But patients can live into middle age, sometimes older, and so to make a decision like that, especially for your child, can be really difficult.
Okay, so we got this gene therapy and gene editing.
Mm-hmm.
Instead of replacing a patient's bone marrow from a donor, you actually modify a patient's own stem cells. Is that correct?
Mm-hmm.
Okay, tell me a little bit about that.
So there, for many, many years, there has been research into gene therapy for many diseases.
Mm-hmm.
And the hemoglobinopathies like thalassemia and sickle cell have been some of the first targets, actually. And in December of 2024, 2 methods of gene therapy were approved by the FDA for sickle cell disease. And, it was very exciting, needless to say, for everyone involved. One method involves adding a gene for a non-sickling hemoglobin
Mm-hmm.
into the patient's stem cells. And the other is a gene editing method where you are actually going in and inactivating a molecule that
represses.
that suppresses the production of fetal hemoglobin.
Okay, fetal hemoglobin is what we all have when we're in our mother's uterus, and at birth we have a repressive gene that stops fetal hemoglobin, and we get regular adult, I guess, hemoglobin, if you want to call it as such. Well, that's very interesting. So it doesn't seem as invasive and difficult as the bone marrow approach, I think. I mean, it sounds like that, but inform me, is it?
It is still a bone marrow transplant. The big distinction is that the donor and the recipient are the same person.
Uh-huh.
So this is what is called an autologous bone marrow transplant.
And it's the patient's stem cells, the individuals suffering from the disease's stem cells, correct?
Correct.
Okay. How do we insert normal functioning hemoglobin genes into a piece of the DNA and allow normal functioning hemoglobin to be made? Something about a virus vector I read, or something like that?
So depending on which FDA-approved product you're using, one of those products uses gene editing, so it uses a CRISPR-Cas9
Mm-hmm.
model to edit the expression of DNA to turn on the expression of hemoglobin F, which
Mm-hmm.
does not sickle.
Mm-hmm.
And the other uses a lentiviral vector to insert or add a functional hemoglobin
So
A.
you insert the normal stem cell into a virus that's harmless, correct, or no?
The other way around.
The other way around.
These genetic products are inserted into the stem cells. So the stem cells are taken out of a patient's body. That typically requires several cycles of apheresis. So
Okay.
normally your stem cells live in your bone marrow.
Mm-hmm.
Patients with sickle cell disease, when they receive growth factor, tend to go into vaso-occlusive crisis, pain crisis.
Mm-hmm.
And so they don't receive growth factor to make their stem cells leave their bone marrow. Instead, they get a different kind of medicine that causes a slower release of those stem cells. It's not as powerful a push.
And they're easier to get or
they're
to harvest.
Easier to get, but it typically takes longer to collect adequate stem cells for gene modification from a patient with sickle cell disease than it would take to collect stem cells from another patient, for example, a cancer patient.
Understand.
So our patients on average get anywhere from 1 to 3 cycles of collection that occur over the course of months.
Gotcha.
Once we've collected adequate stem cells, all of those collections are frozen and then sent to the pharmaceutical company for genetic modification.
Wow.
They're genetically modified either using CRISPR-Cas9 or lentiviral vector, tested for quality and safety, make sure that there's no infection, there are no concerns about the product itself, refrozen, and then sent back to us.
Okay. You mentioned the CRISPR method of gene editing. Now, the CRISPR method, I'm going to tell you what CRISPR stands for. It's Clustered Regulatory Interspaced Short Palindromic Repeats. A big word there, a big mouthful there. Explain that as best you can. CRISPR. What is CRISPR?
So it's a relatively new technology. We've been using the older technology for far longer.
The ones we talked about already, right?
Correct. Where a certain segment of DNA is identified by the CRISPR and then sort of latched onto and then edited. And in this case, what's been latched onto is the repressor of
fetal hemoglobin.
F.
Right, fetal hemoglobin. Interesting. Now, you have a scissors too, right? Cas9 or something like that. Or that's the scissors that cuts the gene?
Yep. That's what we refer to by editing. So,
Interesting.
And if you think about a traditional film editing or recording editing, you think about someone taking a pair of scissors and then cutting out what they want to cut out and then gluing things back together.
And I understand that repressor gene that's holding back the formation of fetal hemoglobin is called BCL11A?
Sure.
And you call the scissors, why do you call the scissors what is it?
Cas9.
Cas9, yeah. Why is it called that?
It's
just curious.
It's an enzyme, so they're not actual pair of
Yeah,
scissors.
I understand that. I understand that they're
molecular
not
scissors.
But they're
Yeah.
molecular scissors.
Yeah.
That's okay. Good way to look at it. Oh, wow. That's really interesting. This is the future in many, many disease entities that we have the potential to cure or reverse or lessen symptomatology. I have patients with Huntington's disease. It's a genetic neurologic disorder that is devastating to the human body, and it's horrible to see. This could work for that, right? Not sickle cell here. We're talking about another disease entity. Can we do the CRISPR on the Huntington disease patient in time?
The short answer to that is I don't know, because I don't know enough about Huntington's. But what I can say is that one of the reasons that sickle cell and some of the other hemoglobin problems were the first to be successful is because they're caused by a very small change in the DNA.
Okay.
It's not, caused by something that's modifying the DNA. It's caused by a single base pair change. So it can be more easily targeted with gene therapy than some of the more complex molecular diseases where you have, genes that are flipped or genes that are crossed over with other genes.
or additional copies of genes.
Yeah.
I think the other part of what makes the hemoglobinopathies amenable to gene therapy is that you can take out the bone marrow stem cells, which are the source of all of your blood cells in your body, and then put them back in.
And just substituting the, getting rid of the regulator to allow fetal hemoglobin to be made in the bone marrow. Wow. So, but the potential is there. You said, I don't know because I'm not really familiar with the genetics findings they are finding from hereditary diseases like Huntington's disease. But do you see it as a possibility?
Yes.
Okay.
And I think there are other genetic diseases that are being treated with gene therapy.
Like?
Severe combined immunodeficiency.
Severe combined
Some
immunodeficiency, which
HIV.
is SCID.
HIV?
HIV, no.
Okay.
Hemophilia, some rare childhood genetic disorders, because we're pediatricians, I think we're more familiar with those disorders. And other disorders that primarily affect other organs in the body, like the liver.
Okay.
For example, hemophilia is caused by deficiency of a protein that's made by the liver. And there have been gene therapies developed to target those cells.
So you look for the hemophiliac sequence. Is there such a thing? A sequence in the DNA that leads to hemophilia? Or are there multiple sites in the DNA that leads to hemophilia, as far as you know? Good question, I guess.
So there are different types
I've baffled
of
the experts, everyone.
There are different types of hemophilia.
Yes, I understand that.
And so, in general, each one of those gene therapies target those specific genes that are mutated to cause that disorder. And it's complicated.
I'm sure it is. It doesn't sound easy.
I think one of the big concerns in gene therapy is causing no harm.
And in medicine in general.
So you don't want to potentially accidentally target an off-target gene and modify that off-target gene because of the concern that you might end up with another disorder. A big concern is the risk of a secondary cancer.
I understand that.
And so that is why these studies, even though these technologies have been around for a while, that's why these therapies have been sort of slow to hit the market because of this concern that you don't want to cause another disease or another problem in exchange for the primary problem.
But the future, I feel, is bright because we at least know the DNA structure and that we can find the gene of whatever causes whatever and address it somehow, some way, either through the first method that we talked about or the CRISPR method. This has been really fascinating, I got to say. You guys are really doing some good work, and it's it's greatly appreciated. How are you doing statistically with all of this? I mean, how many patients have you treated? How many have gotten better? God forbid, how many have gotten worse? Do you have any of that data? I think we could find a time here to take a break and come back with some interesting questions I want to answer. But our break is to identify our sponsors again and to get some information from our public on how we can improve or give us ideas that we can use in our future podcast.
Information about the Medical Society of Delaware can be found on our website, medicalsocietyofdelaware.org. Questions about our podcast can be emailed to healthcare.reality@medsocdel.org. That's medsocdel.org. Once again, here's Dr. Nick Biasotto.
back from our break. This is Dr. Nick Biasotto, host of Delaware Healthcare Reality, a podcast of the Medical Society of Delaware, with our two guests today, Dr. Robin Miller and Dr. Emmy Caywood. We've talked on this subject, which to me is fascinating and very interesting, and I'm sure it is to our public that are listening. I know that you've treated people with either of these methods in the past. Your experience is great. Tell me some of the statistics. How have you done with these procedures? Have they really helped people? What percentage it didn't? What percent it did, if you have that data?
So we're talking about the big we, not the Emmy and me we.
The big we.
The big we, because most of the data that we have so far is from the clinical trials. And it's outstanding. I mean, better than 95% of patients who've undergone both therapies have had a reduction or complete absence of severe pain crises, which was the main marker that they were measuring, and also resolution of their anemia. Patients with sickle cell are very anemic at baseline. Resolution of their hemolysis or red cell breakdown. So it looks like a cure, but the problem is we don't know if it's going to be durable forever.
Gotcha.
And we're not going to know that really for many, many years. It's taken quite a while after the approval to get the therapy to patients off clinical trials, partly because the companies had to ramp up to be able to manufacture the cells, and partly because it can take up to 6 months or a year to be able to collect enough cells and get patients ready to undergo the treatment. So each institution is starting with just a couple of patients. We have 3 patients, I think, now in various stages of going to get their cells soon and have undergoing the collections. But it's going to be really, really important to collect data over many years so that we know what the risks are, really. We know the short-term risks from the clinical trials, and there have been probably a couple hundred patients total treated so far. The farthest ones out are maybe 12, 15 years now.
I was thinking on average 10.
Yeah, definitely on average less than that.
And they're doing okay
so
at
far,
this
so
point?
good,
Wow,
for
that
the
sounds
most
good.
part. But some of these risks, like secondary leukemia, can come many years later. And also, we don't know for sure whether the durability of the treatment is going to be lifelong. We hope so. It seems like there's a good chance, but those are some of the uncertainties that the patients face.
Okay, say the sickle cell patient, the family comes in, they're talking to you, you offer 2 types of approach to their condition. Do they pick the CRISPR or do they pick the bone marrow destruction method?
I would actually take a step back.
Okay, step me back. Go ahead.
And I still think bone marrow transplant, or traditional bone marrow transplant, is still on the table as far as a choice is
Oh, it is,
concerned
but I'm
for
saying
curative
if you
therapy.
offered that to them, which do they select? I'm just curious. Do they have an option?
They do have an option, and I think we as a community are trying to sort the answer to that particular question out.
Okay.
The reason why a traditional bone marrow transplant is still on the table is because we have about 40 years of data
Experience with that.
in pediatric patients who have aged to be adults, middle-aged adults. So there is a proven track record using a bone marrow transplant.
CRISPR is rather new.
CRISPR is rather
but potentially revolutionary, yes?
Yes.
Okay.
And the major advantage of the gene therapy products is that you don't have to go looking for a well-matched donor.
Right.
Only about 15% of patients with sickle cell disease have a completely matched donor. And for that reason, bone marrow transplant has had limited availability to the patient population that we're talking about.
Gotcha.
So even if you met the severe criteria to undergo curative therapy like a bone marrow transplant, if you didn't have a donor, you couldn't go past go.
Now, before you initiate the destruction of the bone marrow first through chemotherapeutic agents, do you have a donor? Or do you have them sitting there and waiting for a donor in a room that's sterile?
No.
You have the donor.
The donor is identified. The donor is cleared. So every donor has to undergo testing, not only to make sure that they're physically well enough to donate, but also emotionally well enough to donate.
You have to be emotionally set for this, yes.
So every donor undergoes that clearance before a patient starts their prep. If a donor is not cleared, the patient doesn't proceed.
Understand.
And for gene therapy, we wouldn't give the chemotherapy
Correct.
to wipe out the bone marrow until we received the cells and back from the pharmaceutical company that had been modified, and they've been checked and cleared.
What pharmaceutical companies are working on this? Do you know?
There are two with FDA-approved products. One is Genetics, previously known as Bluebird Bio. They underwent a name change about a year ago.
Okay.
And then the other is Vertex.
Okay.
And they're the owners of the CRISPR-Cas9 product.
Right, nice. Anything else?
This is really expensive therapy.
That's a good point. How much?
It costs now between $2 and $3 million, depending on the method that's used, just to prepare the cells. That doesn't include the cost of the chemotherapy and the hospitalization and all of the management that goes around that.
I sure wouldn't want to go for a prior authorization for that. That's for damn sure. Oh,
Yeah.
excuse my French.
So far, we've been able to get approval for the patients that we've recommended for gene therapy, but you can imagine this is not going to be something that's available to sickle cell patients around the world anytime soon, unless the process is made a lot less complex and a lot less expensive. Even patients in this country certainly won't all have access to it in the current
Have
situation.
traditional commercial insurances paid for some of these?
Yeah.
They have?
Yep.
God bless them.
Yep. We're not flying patients in because right now we've got plenty of our own. And again, we're pretty limited in how many you can do at once in one institution. There's institutions around the country who are approved to do this. We would fly someone in if they wanted to come, and
Flight's nothing compared to $2 million bucks,
I would say, though, that there aren't separate lists for an institution in Wilmington, Delaware versus an institution in Seattle, Washington, because really the rate-limiting factor is the ability to produce the genetically modified cells. So that's a singular wait list. So it doesn't make a difference if you are living in, Denver, Colorado, if you get in line in Denver versus you get in line in Wilmington, Delaware. All of those lists meet at the same place.
Well, let's talk about the geographic possible implementation. Do people come from across the country? Are they able to get in line for a bone marrow transplant? Is it difficult to do? Is it adding to the cost of the program? Is it working?
So there's centers all around the country doing transplant for sickle cell disease. And there are also a smaller number, but centers all around the country who've now been approved to do gene therapy. And in general, the centers that have been approved for gene therapy are centers who already do have pretty robust experience doing transplant for sickle cell disease. And, Emmy is more than familiar with.
Well, you mentioned earlier there are 100,000 sickle cellers in the United States. Is that true? Yes. So are you reaching them all, or are you reaching 10%, or
not even
close?
Not even close. Again, each center can only start with a couple of patients, and that's kind of where we are right now.
Wow.
I do think that as we advance, we will expand our ability to treat more patients. The other interesting, I think, really interesting part of this field is we've optimized a lot of different things around the preparative regimens, the support of patients during the course of their actual transplant or gene therapy, such that outcomes are pretty good, but we still have a ways to go.
Understand.
And with gene therapy, I think there's a huge potential for trying to explore avenues to do this kind of therapy without chemotherapy.
Yeah.
Great. That's important to hear.
And right now we're stuck with that because we need the chemotherapy to make the gene therapy successful. But I would expect in the next decade that we can start to walk away.
So more of the CRISPRs.
So more of therapies that don't come with the toxic side effects of chemotherapy. So more immunotherapies and targeted therapies that eliminate just the cells you want to eliminate.
Wow. Wow, this has been fascinating. A very interesting discussion, both of you. I'm glad somebody told me about this. It was one of our members said, you got to talk to these people about gene therapy. And I said, okay, but I got to brush up on it, obviously, before I go in there. Anything else you would like to add? Anything you could see as possible support from the medical community to help you or to, participate in any way? So the question is, can you give us some tips on how to possibly be helped by our medical community with your research, and how can people learn more about what you do? That's important.
I think one of the most important things about these new therapies is have your patient talk to an expert, because the field has advanced quite a bit. When I first got into this field 20 years ago, transplant and sickle cell disease was associated with a very high mortality rate, which is why, in part, it was reserved only for the sickest patients with sickle cell disease. Fast forward to 20 years present, that has changed significantly. We are now doing transplants with no chemotherapy. We're doing transplants with low doses of radiation and immunotherapy, much more limited toxicity and mortality with transplant. And now we've added gene therapy, which opens the window to transformative therapies for patients without a well-matched donor.
Gotcha.
And so if you have a patient with one of these conditions, it's really important to talk to an expert about them. They may walk away and say, that's not for me, but I think knowledge is power, and to just understand what the options are out there is valuable. That's part of the reason why Robin and I are now working so closely together, is to provide that kind of consultation to patients here in Delaware.
Well, you guys are the experts, so how do we get them in touch with you guys?
So our website, we're both on faculty at Nemours Children's Hospital in Wilmington, Delaware. Our website is nemours.org. Nemours is spelled N-E-M as in Mary, O-U-R-S.org. And we can also be reached.
That's what I'm trying to think.
Well,
Can I add one thing?
yes, sure.
Am I allowed to do that?
Go right ahead.
Just to go back to your last question, what can the medical community do?
Correct.
I think it's critically important for patients with sickle cell disease to be managed at sickle cell centers or to have access to sickle cell centers. And in our country, in pediatrics, that works pretty well. but there are not a lot of adult sickle cell specialists and hospitals that have sickle cell centers for adults. And that's a problem because these therapies, are options for adults. But in order to go through some of these really difficult and curative therapies, you have to have good organ function. You have to have had your disease managed properly all the way along,
Sure.
so that you're going into it in the best health as you possibly can. And you also, like Emmy said, have to get the information. So I think, that's one of the most important things to get across.
In my 46 years in practice, I've had 2 in private in my practice that have had sickle cell disease, but I saw quite a bit of them when I was director of a wound care center because the wounds are horrendous and painful. So I see what it does to people, and it's quite sad. I've lost both of the 2 that had sickle cell that I took care of over the years, and it was way before we even were talking about this kind of stuff. So it's a terrible disease. It really is. This is Dr. Nick Biasotto. This has been a fascinating discussion. I learned a lot. I hope you've learned a lot. And I would like to thank our guests, Dr. Robin Miller and Dr. Emmy Caywood, who has been really opening my eyes to some things that I really was unaware of. And my research to get prepared for this was very interesting, too. And keep up the good work, ladies. I appreciate all that you do. And this, again, Dr. Biasotto saying goodbye till next time.
Thank you. Thank you.
You're welcome.
This has been Delaware Healthcare Reality, a podcast presentation of the Medical Society of Delaware. For more information about MSD, visit our website, medicalsocietyofdelaware.org. For questions or suggestions regarding our podcast, email us at healthcare.reality@medsocdel.org. Thanks to Audionautics.com for our music. The executive producer of Delaware Healthcare Reality is Mark Thompson. Delaware Healthcare Reality is produced by Tom Mitten. For the Medical Society of Delaware, I'm Lori Ann Rhodes. Watch for our next podcast episode. Until then, be well. The Medical Society of Delaware, MSD, is providing this podcast for informational purposes only. It provides neither medical, legal, nor any other professional advice. The views, thoughts, and opinions expressed by the host or any guests are their own and do not reflect the views or policies of MSD. Unless specifically stated, reference to any specific product, service, or entity does not constitute an endorsement or recommendation by MSD. Reliance on the information provided in this podcast is exclusively at the risk of the listener. MSD expressly disclaims any and all liability or responsibility for any direct, indirect, incidental, or other damages arising out of any individual's use of, reference to, or reliance on this podcast or the information presented in this podcast.